Welcome to SARC Talk. SARC is a nonprofit pan-sarcoma research organization that develops and manages clinical trials in pediatric and adult sarcomas. SARC Talk is a podcast to discuss activities of SARC, interview sarcoma research leaders, and review innovative scientific initiatives in the sarcoma field.
Welcome to SARC Talk, episode number 4. My name is Scott Okuno, medical oncologist at Mayo Clinic and Chief Medical Officer of SARC. All episodes are posted on our website, Spotify, YouTube, SARC channel, and wherever podcasts are available. Please post comments because we will review those and make adjustments as we go along. Today, our guests are Dr. Laura Davis, Associate Professor of Medicine in the Division of Hematology, Medical Oncology, Oregon Health Science University, member of the Knight Cancer Center, and Dr. Angela Herby, Assistant Professor, Department of Medicine, Oncology Division at Washington University in St. Louis at the Siteman Cancer Center. Welcome. It is great having you both on our podcast today, and we look forward to having discussion.
Thanks for having us.
Thanks, Scott. Good to see you, Angie.
You too.
There are some common themes that I did pick up as we were getting ready for this podcast. A couple of the themes I did pick up is that you both were SARC CDA awardee winners in 2016, so we'll get into that. You both practice AYA, so we'll like to hear a little bit more as how that has impacted your practice. Both of you have developed a sarcoma experience with excellent mentors, and I think that's a key part that we're trying to pull through some of our SARC talks as well. And the other aspect that's quite fun to watch your careers is that both of you have developed leadership positions nationally, and how does that impact sarcoma community and SARC? So with that, Dr. Davis and Dr. Herby, please describe your roles at your institution so people can get an idea. Maybe we'll start with you, Dr. Herby.
Okay. I am part of our sarcoma program here, obviously, but also wear a second hat in terms of caring for cancer predisposition syndrome patients. And I also have a basic science research lab as well, where we're interested in using genomic studies of our patient tumors to try to identify novel biomarkers to help in early diagnosis and also to try to develop more targeted therapeutics. So from a clinical standpoint, I'm in clinic, one full day a week, and I see a variety of sarcoma patients, mostly in sort of that AYA population. And then I also see all of the cancer predisposition syndrome patients at WashU and Siteman who have a predisposition for developing sarcomas. So there's a couple of us that see that patient population, and I take basically patients with neurofibromatosis type 1, the Li-Fraumeni syndrome patients with SDH germline mutations. And then I also take some of the FAP population, those patients that have a diagnosis of desmoids. And then one of my colleagues takes the FAP patients who've not had a history of desmoid. So that's sort of my general role here.
Oh, wow. That's quite interesting. So let's see how that compares with Dr. Davis. Dr. Davis, describe what your roles are at your institution?
Okay, so I am both in the Division of Medical Oncology and also in the Division of Pediatric Hematology Oncology, actually. So dual appointed as we kind of get to chatting about our AYA connections. I am the sarcoma program director here at Knight Cancer Institute at OHSU. And I also am co-leader of our Cancer Institute's Translational Oncology research program. And all that comes down to I have a lot of meetings, and I see patients actually about 2 days a week. Not much on the way of inpatient coverage, but I do see a good number of folks in the clinic. My subspecialty, like Angie is a subspecialty with MPNST and predispositions. Mine is osteosarcoma. And so I'm actually super interested, Angie, in learning more about your predisposition work, because I do a little bit of that too. So we are like, we got more stuff in common than we even realized. I love it. And so, yeah, as far as the translational oncology aspect of my role, I do a little bit of translational research focused on osteosarcoma, and then in a leadership position, really trying to get folks connected in cancer center members as they are developing IITs and connecting with lab researchers. So it's a busy and diverse job, kind of like taking care of sarcomas, busy and diverse.
You both, it's amazing how your careers both started kind of roughly at the same time, and how even though you might not interact a lot, you've still kind of connected with the same type of goals that you guys started out back when you started. I think one of the things that's interesting for me looking from the outside is, most of us as sarcoma doctors only see sarcomas. We don't deal with the predisposition or the pre-evaluation. And I just like to hear a little bit more of how does that play differently than someone who is newly diagnosed with sarcoma or going through sarcoma management, and how does that influence you and your discussions that you have with patients with sarcoma? Maybe we'll start with Dr. Davis first.
Oh, yeah. So as far as predispositions, this is kind of a role I've fallen into because we're kind of in the early stages of developing a robust predisposition clinic. And so Lee Fraumeni patients I was seeing as a sarcoma doctor, of course, and realizing that many of them were not getting the recommended cancer surveillance and screening. And so I have been following Lee Fraumeni patients now, in particular with whole body MRIs, and really trying to emphasize the importance of identifying cancers that are extremely likely to show up at some point for us to be able to find them earlier. And if we find a cancer early in someone with one of these cancer predisposition syndromes, then if it's a sarcoma, I will be taking care of them and making sure that they're getting the and not getting radiation and things like that. And with other cancers, of course, if they're developing a colon cancer, make sure they're going to the right specialist for that. I'm fascinated by patients with NF1, with neurofibromatosis. And of course, Dr. Herbie is really the expert there. And I will absolutely be reaching out to her as we develop our NF1 surveillance clinic or make it a little bit more robust.
So Angie, any additional comments? Because it sounds like your path, you're focusing on predisposition for potential sarcoma cases as well. And it dovetails with what Laura said as well.
Mm-hmm. Yeah. So like Laura, I sort of initially fell into that role because there was a bit of a gap here. We had a really strong cancer predisposition clinic on the pediatric side. And they really needed somebody on the adult side to help with that, which is how I fell into that role. And then luckily we had recruited somebody else here who's interested in cancer predisposition syndromes, who wanted to see, some of the patients that will not probably get sarcomas, because it was a little much to try to hone in on all cancer predisposition syndromes. And so like Laura said, if these patients get sarcomas, I'm the one who will treat them. What is different about this population than, seeing somebody up front when I have a diagnosis of sarcoma and I'm, going through how we're going to manage this tumor, is that, because I'm specialized, when somebody gets a different type of cancer, I can't give them that plan right away. And so that can, I think, sometimes be a difficult conversation, because I think one of the hardest things for patients to deal with is uncertainty. And so, I will sometimes have to tell them that, there is a cancer here. I can't tell you the plan right now, but I'm going to get you in to see the right person so that you can have a plan.
You're right. And I think, Laura, you said that too, is if you find a tumor that's outside your area of sarcoma expertise, it's making sure that we have this warm handoff but here you've followed these people for a long time. You feel like you're not providing good care, but you're probably providing the most appropriate care, handing them off to experts that do deal with that. And clearly making sure that when decisions are made in that treatment, that they don't do things that potentially will harm the patient long term. So it's interesting how you both have come to that part. And that gets back to 2016. Both of you were awarded a SARC CDA. And I like to think about your career paths. And what did you think you were going to do with that SARC award? What did you accomplish? And what advice would you give to new awardees? Because we're getting through a new set of awardees, and we're trying to share wisdom that you guys have had. So maybe you can say, hey, what you thought you were going to do, what did you do, and what advice?
So what I thought I was going to do, I think I was using it to get my lab started off the ground, and I think that was a successful endeavor. So I had the unfortunate luck of being diagnosed with breast cancer at the end of fellowship, and so my plans to submit a KO8 were derailed. But by the time the SARC CDA application was due, I was sort of in a better space, and so submitted what I planned for a KO8 for that SARC CDA. And so luckily I got that. And so that was really the grant that was the foundation for me starting my own independent lab, and sort of was able to have that protected time and the ability to hire a lab manager who's still with me to get things going to a point where I could get something else that, more grant funding, and sort of, get the ball rolling and keep the lab going. The exact project that I was working on for the SARC CDA has not panned out the way I would have hoped. But it was that research that sort of led us down the avenues that we've gone, which kind of leads to my advice of follow the science, because again, the actual project didn't work out exactly the way that I thought it was going to, but it still led us down, avenues that were important to follow.
Thank you, Angie. And so those that are listening, Angie's doing well from her breast cancer sample. So just make sure we know that, okay? But there's detours that happen in her career, also in her lab and her project, and that's a very good advice that you have for individuals. Laura, how about you?
So the Career Development Award was huge, and the project, I think this is also a theme. The project did not go according to plan. I also feel like that is pretty standard for lab work, right? Like I had this amazing, it was all planned out. It was all, these are the experiments. This is what's going to happen. Here are the pitfalls. I'm totally got it all figured out. Of course, none of that actually went according to plan. But the bottom line is that I am still 100% in sarcoma research. So it absolutely did the job of developing my career. So the award protected time for me to spend my time, my brain energy on sarcoma. And so I think that was highly effective. Me with a pipette, not so successful. And actually, sometimes I feel a little like I didn't do what I was supposed to, or something like that, as a, because I didn't stay with the lab research. Essentially, the lab research that I proposed in the CDA, I did that work, and then I clung on to the idea that I was going to be able to have a lab for many years after that. But the fact of the matter is I'm not that good at it, and it's not my passion. And so what I've come to now is I love to partner with folks who know the assays and the analytics inside and out. And I absolutely will be able to figure out what the mouse experiment should look like, and when we should get the tissue and what drugs we should use for the mice. But I am not the one to be actually taking those biopsies and looking at that, right? So what I can do is bring that into clinical trials and connect with the right lab researchers, and that early work really helped me identify the things that I was good at, as well as the things that I was not good at, and helped me find my niche.
As we look back at the goal of the CDA, it is to help support and fund people that will be committed to a career in sarcoma research. And both of you are living proof of that, both from the lab standpoint, but also the connector lab person as well. So I think we have to reflect and say, yeah, those are successes that both of you guys have had in 2016. And for people that struggle with, say, hey, it's not the right thing to pipette and do biopsies, it's okay. And not feel bad, because you're still engaged in sarcoma community, and that's the key part. You both work in the AYA, and I think it's an important aspect. And I know we at SARC try to do clinical trials that includes and ensures that we do include the AYA population. I'd like to hear your thoughts on the value of AA research, because this is a little bit newer than maybe we had when I started through the practice, and how it impacts your adult practice, because some of us are just adult practicing people, although the AYA does cover that part. So how does the AYA impact your practice, let's say?
So AYA was actually part of my life and training before sarcoma. So I did a MedPeds residency and a MedPeds combined fellowship in order to focus on adolescent and young adults with cancer. And so I started out there, and then it became quite clear that sarcoma was a big chunk of that, right? So although sarcomas are super rare in the population in general, 1% of all cancers, right? But it's 15% of pediatric cancers, and I find it really interesting that probably about 50% of all sarcomas happen in AYAs. So I took my training in pediatric oncology and combined it with my training in medical oncology, focused on sarcoma patients, and in particular, young adult patients and adults that develop quote unquote pediatric type sarcomas. So I actually only see patients on the medical oncology side. I don't see patients on the pediatric side, in part because there's so many more sarcomas on the adult side. But I generally will be seeing the sarcoma patients that are in that young adult category. I will be seeing the patients with osteosarcoma, Ewing sarcoma, alveolar and embryonal rhabdomyosarcoma as a subspecialty. And I love it. I love having that training and interest and connection to the children's oncology group, and bringing the pediatric type regimens to adults, and part of the clinical research that I've done over the years has been looking at things like, can we do interval compressed VDCIE in adult Ewing sarcoma patients, or can we give high dose methotrexate to adults with osteosarcoma? And turns out yes. Carefully.
I was going to say, they don't bounce back as well as children do.
Amazingly, it actually turns out that the interval between doses for interval compressed VDCIE, at least in our cohort, was the same as with the pediatric publication. So it's 17 to 18 days. It's not 14 days, but it's not 14 days in kids either.
Thank you. Angie, your input on AYA.
I think this is so. I'm unlike Laura. I'm not actually trained in pediatrics. So, but we again partner really closely with the group at Children's Hospital, and I do have a joint appointment in pediatrics, mostly so that we can manage trials effectively. I would never attempt to treat anybody who was not an adult-like teenager, though. So I think this patient population just has a lot of unique challenges, and it always reminds me that the challenges that patients face are very different across the lifespan. And things are just intense, I think, in this group, right? It's like the time in your life when you're realizing what you want to do and you really think about the future, and having a diagnosis of cancer kind of can shatter that to some degree. But then there are challenges that children face and older adults face as well. So it just sort of, I don't know, I feel like this is like an amplified population. And it just reminds me to be aware of all of these other things outside of cancer treatment that impact how people deal with their cancer treatment.
You both highlight aspects that I was thinking about as well. One is taking the mindset of a patient. And the mindset of a patient in the AYA is different than someone who is 60, 70 years old. And having that understanding is key. And then the other aspect is taking the types of treatment that we use in the pediatric population and seeing how that would work in the adult older population when it could be beneficial is great. And I think having both of those perspectives that both of you have shared is key for us, because sarcomas, as you said, 50% are in the, so it's at the 40 or under group. I can tell you most of our patients are in that group. Yes, we do get the high end part, when you're 60, 80, but a good portion of our patients in the adult world are there. And having that perspective, the AYA training and collaboration is going to be key. So it's interesting how both of you have come through that, and it's probably lent itself really, really well to in sarcoma. As we think about that in how we do these collaborations, both of you have collaborations outside of your institutions. And maybe, Angie, I know you do work outside your institution. Maybe you can share with you what you do outside of your institution nationally and collaboratively.
I think that's one of the things I love the most about science is team science and collaboration for multiple reasons. One is I think it makes it more fun, and two is I think it makes it more productive because everyone has unique talents, and I think working together we can get far more done than trying to work, independently. And so I have two kind of major collaborations that are, multi-institutional. So one of them involves kind of close collaboration with groups at the National Cancer Institute. So Brigida Wiedemann and Jack Shern and Taylor Sunby there. And then another kind of internal collaborator, Otto Chowdhury, where we're trying to use cell-free DNA technology to try to come up with diagnostic tools to help in early diagnosis of cancer, predominantly in the NF1 cancer predisposition population, but could be, tailored to any cancer predisposition population. And again, more together than separately, right? Because this work would be so slow if we weren't working with a bigger group to have enough samples to ask meaningful questions, and different expertise that we all bring to the table. Similarly, I have an ongoing collaboration with Christine Fertilis at Johns Hopkins, Dave Larga-Spada and David Wood at University of Minnesota, and then Sarah Goslin at PNNL. And that's sort of bringing together sort of bioengineering, bioinformatics, true pediatric oncology, adult oncology, and genetics, really trying to develop novel platforms to test drugs for sarcomas. And then, in novel kind of in vivo models, which that work we hope will lead to, clinical trials that somebody else like Laura can run. Because what I have learned is that While I am great at having a lab going and taking care of patients, the idea of actually running a trial myself is probably not the best idea. Too many balls will be juggled. That it definitely requires partnership.
Laura, collaborations outside your institution.
Yeah, so I agree with Angie that the collaborations, the team science, whether we're talking clinical science or laboratory science, is fantastic. And as all of us know, everybody who's listening knows, the sarcoma community is fantastic, right? We are so collaborative, so fun. And I'm so fun. We're all so fun. but it's a small group of patients. It's a small community of people who are researching in sarcoma, and so we have to collaborate. But thankfully, we like to collaborate. I think kind of my biggest collaborations are actually through the large national groups. So SWOG is one of the NCTN Collaborative Clinical Research Network. And I lead the sarcoma working group within SWOG. And that is a great way to really start to foster clinical research through the NCTN mechanism across the country that focuses on sarcoma research. And then also, of course, I'm very involved with SARC. And I think one of my favorite collaborations that I'm doing right now is with SARC 38, which is our clinical trial for osteosarcoma, advanced osteosarcoma, utilizing an oral multi-targeted tyrosine kinase inhibitor, or regorafenib, combined with checkpoint inhibitor nivolumab. And that clinical trial has a ton of that. So developed and run with Fariba Naveed at CHLA. So that's kind of the biggest, closest collaboration between her and I. And then the trial itself involves a large number of correlative studies. So biopsies and blood draws, archival tissue, and I'm helping to kind of coordinate a collaboration of scientists across the country who will be analyzing that blood and tissue so that we can figure out why some patients benefit a lot and some patients don't benefit at all. So how can we figure out who we should be offering this type of treatment to earlier on and not waiting till third or fourth line? And who should we not be offering it to at all because it's not going to help? That's, I think, such an important aspect of clinical research, is making sure that not only the patient on that trial at that moment is getting the most benefit possible, but also that the next patient on the next trial has benefits from all of the information that has been gathered from prior trial participants.
Yeah, and that brings up, the part that I for those that are listening, I can see Angie's and Laura's faces when they talk about collaboration. And I can tell you that if you're a young investigator working at an institution and you're only working at your institution, you're missing out, because the reality, there's so much richer collaborations in the sarcoma community that will help our patients out. And Angie, I forgot all the collaborations that you had. Then I started to realize, and some of those people I do know too. And now I know that how important it is. And Laura, leading SWOG and the working group there to connect a lot of institutions that are interested in sarcoma and fostering and developing industry to help support that makes a sense. And as well, Laura, you and Sandra leading the concept review committee in SARC is critical. And also being a partner in SARC 38, like you said, I think we've learned a lot. And we're going to learn a lot as we go along. And having both of you involved in the sarcoma community is going to be great. So I'm looking forward to it. both of you came in with great mentors. And I know, Laura, you've worked with Chris Ryan there, and I know, Angie, you've worked with Brian Van Tyne. And I just want to know is that as you now start to develop your careers, what did you learn from them and continue to learn from them? But also now, what are you doing to be those mentees for the new people coming through in your group? Because I know you do have some younger people in your group as well, or at least trying to hire more people too. So, Angie, we'll start with you and how all that mentor collaboration happened at Siteman.
Yeah, I think mentor support is so key. And a lot of times multiple mentors also, right? So Brian has been an amazing clinical mentor to me. I also have, David Gutman as a sort of scientific mentor here in sort of the NF1 space. And then as part of kind of other grant funding, had developed sort of a whole mentorship committee. And again, sort of bringing in different expertise is important. So Brigida Wiedemann is actually kind of on that mentorship committee. And then Tim Lay here as well, who very renowned for genomics work. So I think, my advice is to take advantage of mentorship opportunities, so develop relationships with your mentors and make sure that you find mentors who are, available to you. Lucky for me, Brian's right down the hall, so I can, usually find him. And if he's not on campus, he's usually just a text away. Hey, where are you?
No, that's true. Yeah, I forgot about the idea of the multi mentor, depending on the role they have, and getting that is key. That great insight. Laura, how about you?
Yeah, it's funny. I hadn't thought about the mentorship committee members as a specific group, but when I actually had the SARC CDA having that mentorship committee, the members included Larry Baker, who connected me with SWAB. It included Jonathan Fletcher, who connected me with the SARC Scientific Steering Committee. And of course, it included Chris Ryan. So don't tell him, but Chris Ryan is a really good mentor. I actually, I think prior to having Chris as a mentor in fellowship and then as a junior faculty, I don't really think that I'd had true mentor who helped me advance and figure out stuff, right? Chris on that CDA mentorship committee, he doesn't do lab research. He does clinical research, and he does, sarcoma clinical care, and he's excellent at it. And so he'd listen to my presentations about the Western blots and the cells dying and et cetera. And yes, great. But then he was like, well, I know this person at this drug company. Why don't you pitch a trial concept to him? And that, coming directly out of those experiments. And so I did. And I had my first investigator-initiated trial coming directly out of that. So it really is. It's not even that formal mentorship relationship, right? It's the championship. It's the, hey, talk to this person, or hey, why don't I set up this meeting for you, or hey, why don't you take over the PI of this national trial because you're really interested in osteosarcoma, and I think that'd be a good role for you. And then I'm PI of SARC 24 osteosarcoma cohort, and that sort of championship is really important. And so yeah, it's not. I don't think I even had like regular meetings scheduled with Chris Ryan, but it was all of those little pieces that absolutely helped me get where I'm at now. But don't tell him.
One thing that's unique that I think most of us in the sarcoma community understand is we're all here to promote junior faculty, get them on the podium, get them as first authors, give them national abilities to run clinical trials, because that's our future, okay? And no one's going to generally backstab you in our community, which we all agree, but maybe we're drinking the Kool-Aid too. But the whole goal is, let's get more and more studies for our patients and get people that have expertise, passion out there and do that. And part of what we're trying to do at SARC as well is, if you don't have that, how can we at SARC build those connections, or how do we move forward with that? So I think part of us getting our face out there, our voices out there a little bit more with these podcasts, and more with a clinical trialist approach that we're going to start to move forward with, is going to be key. So I really enjoyed the time we shared here, both Angie and Laura, with what you have. And any additional thoughts or comments that sparked your interest during our conversation today?
Like Laura said, we're a fun and friendly group of people. And I think, the SARC meetings associated with ASCO and CTOS are sort of a great way to connect with people too, especially for sort of the people maybe just getting involved in sarcoma research, or, the new CDA recipients. That was actually the first time I talked to Brigida, was at the SARC meeting associated with ASCO. Yeah, I think, again, this is a fun community.
Thank you.
Yeah, I would second that, and make sure that when you're at a SARC meeting, that you come find Dr. Herbie or myself and introduce yourself, and see how we can help.
We appreciate it. Well, Dr. Herbie and Dr. Davis, thank you for a great SARC talk. Please share SARC talk for those that are interested in sarcoma, because this is the way we can share our message. Please leave comments and suggestions on our SARC Talk podcast, and we'll address it at our next SARC Talk. Any questions that do come up. And we look forward to our next SARC Talk in March of 2023. So thanks, everybody.
Thank you for listening. Be sure to subscribe so you never miss a SARC Talk. To find out more about SARC, please visit our website at sarctrials.org. To suggest a topic or ask questions, please email us at sarctalk@sarctrials.org. And remember, together we can find a cure for all forms of sarcoma.